Parkinson's disease and related syndromes are caused by a malfunction of an area of the brain called the nigrostriatal pathway, billions of individual cells called neurons. These consist of a central cell body that contains all the structures of metabolism and function. The fine tendrils emanating from the cell body are called the dendrites and can carry information either to or away from the cell body itself. The dominant extension off the cell body is called the axon. The axon is covered by a series of sausage-like tubules called the myelin sheath, an insulating structure composed predominantly of fat, cholesterol, and protein that facilitates the rapid transmission of electrical signals either down the axon in motor neurons or up the axon in a sensory neuron. Communication between these billions of cells is facilitated through the synapse, a small gap between the axon of one neuron and, in this particular case, the dendrites of the next. The area before the gap is called presynaptic, and just after the gap is, of course, postsynaptic. The presynaptic cleft contains innumerable vesicles of chemicals called neurotransmitters. When an electrical pulse travels down the axon, it causes these little vesicles to degranulate and spill their chemical content into the synapse. The chemicals traverse the gap and bind to receptors on the postsynaptic cleft. If enough chemical is present, a threshold potential will be reached and cause the next cell to fire, thus propagating the signal on down the line.
A chemical known as dopamine is released from an area of the brain called the substantia nigra. This neurotransmitter is released by neurons onto adjacent cells and into a synapse, sending a signal that enables smooth, coordinated muscle movement. In Parkinson's disease, dopamine release by the substantia nigra is diminished. In response, the adjacent areas of the thalamus and globus pallidus interna become overactive, creating a resultant tremor.